Hello, and welcome to my presentation on organ-based visual classification for plant analysis. I would like to thank my teammates, Meghana Kover, Ram Zaveri, Matthew Keaton, and my advisor, Dr. Doretto. First, I will explain some background information to make this presentation a little easier to understand. Classification is the task of assigning a class to a specific image. Fine-grained visual classification is a type of visual classification that is used when the images in your dataset are visually similar. Object detection is finding a specific object or objects within an image. Computer vision is the field that focuses on extracting meaningful data from images and video. And machine learning is the field that focuses on creating and training algorithms to let machines learn and take action without assistance. For this project, we use computer vision and machine learning to classify images of 1,000 different species that were collected by untrained individuals, which means that the images are in varying levels of quality from different angles, zooms, resolutions, and lighting that can make classification more difficult with existing methods. The main portion of this project that I assisted with was dataset creation. As you can see below, we used 5 data labels. From left to right on the figure, they are leaf, flower, fruit, HDL or high density leaves, and stem. The HDL label is used when you have a very dense pack of leaves that are very small, and it's impossible to get individual leaf boxes around each one, so you just use one larger HDL box. And below you can see the number of annotations for each label throughout the whole dataset. After annotating, the dataset was split into 80% training data, 15% testing, and 5% validation. The reason for having both testing and validation sets is to ensure that your model is not overfitting to your data or becoming too accurate on your training data, and then it will not be able to give accurate results when generalized to other data. On the right, you can see a screenshot of our annotation software. We draw bounding boxes around each organ on each image to direct object detection. Boxes are drawn as small as possible to reduce any extra noise in the dataset. And then some images are left unannotated if the quality is extremely low or if it lacks any identifiable features. Once the dataset is created, we move on to training, and we use convolutional neural networks, or CNNs, which are just algorithms that can assign weights and biases to various aspects of an image, and their design was inspired by the visual cortex in the human brain. The 2 CNNs we used for this project were ResNet 18 and ResNet 34, which use 18 and 34 layers respectively. Each layer receives an input, modifies it using a set of equations, and then outputs the modified data to the next layer. More layers mean more complexity, but too many layers can cause overfitting, like I mentioned earlier, which makes your model too accurate on your training data. And this causes your predictions to be inaccurate when you are predicting on your testing data. A confusion matrix, shown on the right, is a method for visualizing results, and it shows ways your model can be confused when making predictions. The one shown on the right was created after training our model on the first 40 leaf species in our dataset. And because there is a diagonal line from top left to bottom right, that means that our model had high accuracy. We predict that our approach will allow classification to work in more real-world applications with less data modification before training. Thank you for listening to my presentation. If you have any questions, please put them in the comments below and I will do my best to answer them. Have a great day.